Hello everyone, welcome to this video where we will guide you step by step through the process of extracting microplastics from fish samples. Stay tuned until the end to find out if Yogeeta discovered microplastics in her river fish samples or not. This work is part of Yogeeta's research and I'm here to document the process and help her. The method itself is straightforward but can be time consuming, so let's get started. The first step is preparing the fish samples. First, identify the samples to be processed from the lot. Second, sort the samples to avoid duplication and ensure a variety of fish types are included. And lastly, wash the selected samples to prevent contamination and make handling easier. Now that the samples are ready, let's move on to the microplastic extraction protocol. The protocol involves 6 key steps. 1. Weighing. 2. Dissection and weighing. 3. Digestion. 4. Flotation. 5. Filtration. And 6. Observation. Step 1. Weighing. The purpose is to gather baseline physical data of the fish and ensure accurate measurements. Procedure. Measure the length and weight of the fish to understand its size in relation to microplastic content. Weigh an empty petri dish or a container to use as a reference for accurate future measurements. Step 2. Dissection and weighing. The purpose is to isolate and weigh the gastrointestinal tract where microplastics tend to accumulate. Procedure. Carefully dissect the fish, ensuring the sample remains uncontaminated.
So I'm using a surgical blade and we'll cut it right under its mouth from here all the way until it's opening here, and we'll remove the gut from this section. So here I go. It's quite slippery.
Remove the gastrointestinal tract, including the stomach and intestines. Weigh the gastrointestinal tract using the pre-weighed petri dish or a container and subtract its weight for precision. Step 3. Digestion. The purpose is to break down organic tissue while leaving microplastics intact. Procedure. Place the dissected gastrointestinal tract into a conical flask.
Now we have to digest the organic matter. For that we will add hydrogen peroxide, which is at the ratio of 20 of the weight that it has. So if it is 1 gram, we will add 20 ml of hydrogen peroxide.
Add 40% hydrogen peroxide at a 1:20 ratio of tissue weight to solution volume. Heat the solution to 60 to 65 degrees Celsius and maintain oscillation at 1200 rpm for 48 to 72 hours to ensure even digestion. Step 4. Flotation. Purpose is to separate microplastics from organic matter based on density differences. Procedure. Add 150 ml of 30% sodium chloride solution to the digested sample.
and it is placed here in magnetic stirrer. It will be here for 72 hours. And then after 72 hours, organic matter and flesh will be digested. We will be left with undigested particles, which will be sometimes soil, sometimes smaller rock particles, and plastics that we will filter here.
Step 5. Filtration. Purpose is to collect microplastics for microscopic analysis. Procedure. Pass the flotation solution through grade 1 Wattman filter paper, which traps microplastic particles while allowing liquids to pass through.
So this filter paper, we need to cut it to the size of the filtration. Almost perfect circle. And from here we take one and we place it here, and then we place the holder from the top and we put the clamp so it holds. The sample goes in. Close it back. Now, if we keep it like this, it will take maybe half an hour or a little bit more, because it works on gravity, so droplet by droplet. To make it faster, we have this motor. We switch it on and it sucks the air from here, the air that is flowing from here. So with the help of that, it makes the process a little bit faster, but it still takes almost 15 or 20 minutes. So we wait until it filters, then we put another one. After filtration, we observe it to identify and measure microplastics.
The final step is observation, where we examine the filtered material under a microscope to identify and characterize microplastics.
And we are using Dino Capture, which is a digital microscope which gives a display on the laptop screen. So if you see here, those are dust particles, and we are going to scan through and we see something over there. So now I'm going to zoom in. So that is what we have. The others, these are all soil particles that was present in fish gut. This looks like a rock particle because it's very shiny, and plastic does not have this kind of character. Plus it is mixed color.
According to Maron et al., microplastics can be categorized into 7 different shapes such as fiber, line, foam, film, fragment, microbeads, and pellet. Their study in the Mediterranean coastal areas identified these various forms in seawater and along beaches. Similarly, Yogeeta has found 4 different types of microplastics in all the fish samples she examined under the microscope, such as fiber, foam, fragments, and pellet. Maroon et al. Also reported frequent sightings of plastic bottles, bags, fishing nets, and food wrappers in marine environments. They hypothesized that hard plastics and outer packaging could be the primary source of fragments, while plastic bags likely contribute to film mainly from broken fishing lines. Additionally, they raised concerns about the toxicity and sampling challenges of fibers, which were also commonly found in Yogeeta's study. This type of microplastic can interfere with both the gut and gills of aquatic organisms, leading to acute toxicity and high mortality rates. Significance of the protocol. This process is crucial for quantifying microplastic pollution in fish, identifying the types of microplastics ingested based on size, shape, and color, and contributing to studies on environmental contamination, aquatic ecosystem health, and the impact on the food chain.
Finished.